Welcome to the Science of Ageless Living. I'm Dr. Ethan Hausman-Marquis, Clinical Director at the Catalyst Clinic, Doctor Scientist, and the only peptide clinic physician in London with a published body of work specifically in this field. This is the Ageless Top 20 Peptides series. Every episode covers one compound, the real biochemistry, the honest evidence base, what we know, what we don't, and what the regulatory picture actually looks like. No hype, no discount codes, just the science. Today, LL-37. And I want to be honest with you from the first sentence because this compound is the odd one out on this entire list, and I'd rather tell you that plainly than dress it up. The last two episodes lived inside the mitochondrion. Humanin and MOTS-c are mitochondrial-derived peptides, tiny signaling molecules encoded in the small circular genome inside your mitochondria, released under stress, studied because their levels track with aging and longevity. That was a clean, coherent story. Peptides that decline as we age, more abundant in those who age exceptionally well, plausibly protective. LL-37 is not that. LL-37 is your body's own antibiotic. It's encoded in the ordinary nuclear genome on chromosome 3, not in the mitochondria. It wasn't discovered in a longevity screen or a metabolic lab. It came out of immunology, out of the study of how our innate immune system kills bacteria on contact. And here is the part that matters most. The reason I'm putting it on the list at all, and the reason I'm being so careful, LL-37 is the one peptide in this series where more is not obviously better, where in fact, more can be actively harmful. It is a genuinely double-edged molecule, protective in one context, pathogenic in another. And if you take nothing else from this episode, take that. So let's do it properly, the same way we do every compound, what LL-37 actually is, what it does at the molecular level, why it cuts both ways, what the human data shows, and what the honest picture looks like, including the regulatory reality, which for this molecule is unusually specific. The basics. Let me start with the name, because for once the name is refreshingly unsentimental. Humanin, you'll remember, was named as a statement of hope. L37 is named for exactly what it is. The mature peptide begins with two leucine residues, L, and it is 37 amino acids long. 2 leucines, 37 residues. LL-37. That's the whole story of the name. It tells you the field it came from. This is a molecule named by protein chemists, not by people looking for a metaphor. LL-37 is a cathelicidin. Cathelicidins are an ancient family of antimicrobial peptides found across mammals. Pigs, cattle, mice all carry their own versions. Humans carry exactly one. LL-37 is the only human cathelicidin. That's worth sitting with. Of all the antimicrobial peptides in this family that evolution has scattered across the mammalian lineage, we got precisely one cathelicidin, and this is it. Now the structure and the processing, because they're the key to understanding both what it does and why it's dangerous when it goes wrong. LL-37 does not start life as LL-37. It's cut out of a larger precursor. The gene is called CAMP, cathelicidin antimicrobial peptide, and it sits on chromosome 3. It encodes a pre-pro protein of about 18 kilodaltons called HCAP18, human cationic antimicrobial protein, 18 kilodaltons. That precursor has 3 parts, a short signal sequence at the front, which directs the protein where it needs to go. Then a large, highly conserved middle region called the cathelin-like domain. Cathelin was originally described as an inhibitor of a protease, and this domain is structurally related to it. And then, at the very C-terminal end, the last 37 amino acids, the antimicrobial business end. That's LL-37. For most of its existence, that peptide is kept inactive and locked away. In neutrophils, the frontline white blood cells of the innate immune system, HCAP18 is stored in granules, packaged up, held apart from the enzymes that would activate it. This separation is not an accident. LL-37 is a membrane-disrupting molecule. If it were floating around fully active inside your own cells, it would damage them. So the body keeps the loaded form and the trigger in separate compartments until they're needed. When they are needed, during infection, during tissue injury, the precursor gets cleaved. In neutrophils, an enzyme called proteinase 3 does the cutting, releasing the free LL-37 peptide. In the skin, a different set of enzymes handles it, serine proteases of the kallikrein family, kallikrein 5 and kallikrein 7 in particular. Hold on to that detail about skin kallikreins. It comes back later, and it comes back as a disease. And it's not only neutrophils that make it. LL-37 is produced by the epithelial cells that line your barriers, the skin, the airway, the gut, and by macrophages. It shows up in saliva, in sweat, in other secretions, and it circulates in the blood. This is a molecule stationed at every surface where your body meets the outside world. Finally, the physical character of the peptide, because everything it does flows from this. LL-37 is cationic. It carries a strong positive charge, a net charge of around +6 at the pH of your body. And when it encounters the right kind of surface, it folds into an amphipathic alpha helix, a helix with a positively charged face on one side and a water-repelling, oily face on the other, a small positively charged two-faced helix. Remember that shape. It's the key to the antimicrobial mechanism, and it's also, as we'll see, the key to the trouble. What it does, the protective side. Let me give you the beneficial biology first, because it's real and it's impressive, and then I'll turn the coin over. The first and most obvious function is direct killing of microbes. This is what LL-37 was discovered doing. Bacterial membranes carry a lot of negative charge on their surface, more than our own cell membranes do. LL-37, being strongly positive, is drawn to them. It accumulates on the bacterial surface, and then that amphipathic helix inserts itself into the membrane, oily face into the oily interior of the membrane, and it disrupts the whole structure, punching it full of holes or peeling it apart like a carpet, depending on the model you favor. Either way, the membrane fails, the bacterium's contents leak out, and it dies. This works against a broad range of organisms: gram-positive bacteria, gram-negative bacteria, some fungi, some enveloped viruses. It even disrupts biofilms, the protective slime communities that make chronic infections so hard to clear. It is quite literally an antibiotic your body manufactures on demand. There's a related trick worth naming. When gram-negative bacteria die, they shed a molecule called lipopolysaccharide, LPS, also called endotoxin, and LPS is one of the most powerful triggers of the septic inflammatory cascade. LL-37 binds LPS and neutralizes it, so it doesn't just kill the bacterium, it helps mop up the inflammatory shrapnel the dying bacterium leaves behind. In the setting of severe infection, that's a genuinely useful dual action. But if all LL-37 did was kill bacteria, it wouldn't be interesting enough to put on this list. The reason it's studied so intensely is that it's a signaling molecule as much as a weapon. It talks to the immune system. LL-37 is a chemoattractant. It recruits immune cells to the site where it's released, calling in neutrophils, monocytes, T cells, and mast cells. And here's a detail I promised you a callback on. It does this largely by signaling through a receptor called formyl peptide receptor 2, FPR2, also known as FPRL1. If that receptor name rings a bell, it should. It's the same receptor Humanin engages. Two completely different peptides from completely different corners of biology, one a mitochondrial longevity signal, one a nuclear immune weapon, converging on the same cell surface receptor. That's the kind of connection that reminds you these systems are more intertwined than the textbook categories suggest. Beyond calling in the troops, LL-37 modulates how immune cells respond to threats. It tunes signaling through the toll-like receptors, the pattern recognition sensors that detect microbial molecules. Depending on context, it can amplify or dampen those responses, and it does something that has nothing to do with infection at all. It promotes wound healing. It drives the migration and proliferation of the skin cells that close a wound. It promotes reepithelialization, and it stimulates angiogenesis, the growth of new blood vessels into healing tissue, partly by transactivating the epidermal growth factor receptor. Chronic, non-healing wounds tend to be low in LL-37, and that observation is the basis for one of the few actual human trials of this peptide, which I'll come to. So the protective portfolio is a broad-spectrum direct antimicrobial, an endotoxin neutralizer, an immune cell recruiter, a modulator of innate immune signaling, and a wound healing and angiogenic factor. On paper, that's a molecule you'd want a lot of. The vitamin D connection. Before I turn the coin over, one more piece of protective biology, because it's the single most practically relevant fact about LL-37, and it connects to something you can actually measure in yourself: vitamin D. The CAMP gene, the gene for LL-37, has a vitamin D response element in its promoter. In plain terms, the active form of vitamin D, 1,25-dihydroxyvitamin D, binds the vitamin D receptor, and that complex switches on transcription of the LL-37 gene. This is one of the cleaner, better established links between a nutrient and innate immunity. It's part of the mechanistic story behind why vitamin D status has been tied to defense against respiratory infection and historically tuberculosis. Sunlight and vitamin D drive production of an antimicrobial peptide that helps macrophages kill the organism. Now, here's where I have to apply the same honesty I apply to everything on this show, because this is exactly the kind of clean mechanism that gets oversold. Yes, in the laboratory, adding active vitamin D to immune cells switches on LL-37. And yes, in people, circulating LL-37 correlates with vitamin D status. But the correlation is clearest in people who are actually deficient at low vitamin D levels, and it flattens out once you're replete. More importantly, when researchers have run the obvious experiment, give people vitamin D supplements, and see if their blood LL-37 goes up, the results have been inconsistent, and some well-designed trials have simply failed to show it. So the honest statement is this: correcting a genuine vitamin D deficiency is a reasonable thing to do for your innate immunity and plenty of other reasons, and the LL-37 pathway is part of why. But the idea that you can reliably crank up your LL-37 by loading vitamin D on top of an already adequate level is not supported. Fixing a deficiency is not the same as flooding the system. The twist: why more is not better. Now I turn the coin over, and this is the heart of the episode. Everything I've described so far assumes LL-37 shows up in the right place, at the right time, in the right amount, and then stands down. The problem is that this molecule, a small positively charged membrane-active immune-activating peptide, is intrinsically dangerous when it's present in the wrong place, at the wrong time, or in the wrong amount. And in 3 well-characterized human diseases, that's exactly what happens. This is why LL-37 is the one peptide on this list where I am genuinely worried about people trying to raise it. Let me take the clearest example first: psoriasis. In psoriatic skin, LL-37 is massively overexpressed, and it turns out that this excess LL-37 does something that flips it from defender to arsonist. When cells die, and there's always some cell turnover and injury in skin, they release their own DNA and RNA into the surrounding tissue. Normally, your immune system is carefully insulated from your own nucleic acids. The sensors that detect DNA and RNA are tucked away inside cellular compartments, kept away from self-material, precisely so that your body doesn't attack itself. LL-37 breaks that insulation. Being highly positively charged, it binds your own released DNA and RNA, negatively charged molecules, and forms tight complexes. Those complexes do 2 things. They protect the nucleic acid from being degraded, and they act as a delivery vehicle, ferrying your own DNA into a type of immune cell called a plasmacytoid dendritic cell, straight to the internal sensor called toll-like receptor 9. The cell reads your own DNA as if it were a viral invader, and it responds by pouring out type 1 interferon. LL-37 does the same thing with self-RNA, delivering it to related sensors on other dendritic cells. The result is a self-sustaining inflammatory fire, an immune response against your own tissue, initiated and amplified by a peptide that was supposed to be defending you. This was shown in a landmark paper in Nature in 2007, and it reframed psoriasis as, in part, a disease of a misbehaving antimicrobial peptide, and it got worse for LL-37's reputation from there. It turns out LL-37 isn't only the messenger, it's also the target. In a large fraction of people with moderate to severe psoriasis, by some measures up to 3/4, the immune system's T cells recognize LL-37 itself as an autoantigen, mounting a direct response against the peptide. So in psoriasis, LL-37 both lights the fire and becomes something the immune system attacks. That's a molecule you would not want to inject into a person prone to the disease. Second example, rosacea. Remember I told you to hold on to the detail about skin kallikreins, the enzymes that process cathelicidin in the skin. In rosacea, those enzymes, kallikrein 5 in particular, are abnormally overactive. And when you overprocess cathelicidin with excess protease activity, you don't just get more of the normal LL-37, you get aberrant, abnormal peptide fragments that are strongly pro-inflammatory. Those abnormal fragments drive the redness, the inflammation, and the vascular changes that characterize rosacea. This work came largely out of Richard Gallow's laboratory, and it established rosacea as mechanistically a disorder of cathelicidin processing. Again, this is a setting where more cathelicidin activity, or the wrong kind of processing, is the problem, not the solution. Third, systemic lupus erythematosus, lupus. The same self-DNA mechanism that drives psoriasis appears in lupus too. LL-37 nucleic acid complexes drive that same type 1 interferon response, and interferon is central to lupus. On top of that, many lupus patients carry autoantibodies directed against LL-37, and they have T cells that respond to it in a way linked to disease activity. So in a second major autoimmune disease, LL-37 sits near the center of the pathology. And I'll add a fourth, more ambiguous thread, cancer. LL-37's role in cancer is genuinely context-dependent and unresolved, and I want to represent that honestly rather than pick the scary version. In some cancers, ovarian, breast, lung, have been reported, LL-37 appears to promote tumor growth, plausibly through those same growth and migration signals it uses in wound healing, acting via FPR2 and the EGF receptor. In other cancers, particularly some gastrointestinal ones, LL-37 or its analogs appear to do the opposite and suppress tumor cells. So it's not a simple LL-37 causes cancer story. It's this molecule is a powerful modulator of cell growth and survival, and the direction of that effect depends on the tissue story, which is, if anything, more reason for caution about pharmacological dosing. A molecule that pushes cell growth and migration is not one you want to flood a body with casually. So here is the honest synthesis of the biology. LL-37 is a superb acute defense molecule when it's deployed correctly, a self-limiting burst in the right place that clears an infection and then stands down. It becomes a driver of chronic inflammatory and autoimmune disease when it's chronically overexpressed, abnormally processed, or bound to your own nucleic acids. This is why the framing that works for Humanin and Clotho, levels fall with age, so raising them might help, is the wrong framing for LL-37. This isn't a dial you want turned up. It's a system you want working properly. What the human data actually shows. Now let's talk about the human evidence. And here the picture is different from the mitochondrial peptides in an important way. With Humanin and MOTS-c, the biology was well worked out, but the human interventional evidence was close to a blank page. With LL-37, the endogenous human biology is even better established. We know a great deal about what your own LL-37 does inside you, but the evidence for LL-37 as an administered therapy is thin and narrow, and the evidence for it as an anti-aging or general immune injection is, for practical purposes, absent. Let me take the endogenous side first, because it's strong. We know LL-37 matters for human host defense because of what happens when people don't have enough of it. There's a rare condition, a form of severe congenital neutropenia, sometimes called Costman's syndrome, in which affected individuals are deficient in LL-37 and they suffer from severe recurrent infections, including devastating periodontal disease. More broadly, low cathelicidin is associated with increased susceptibility to skin, airway, gut, and urinary infections. So the case that your own LL-37 is important for keeping you free of infection is solid, based on human genetics and human disease. That part is not in doubt. The trouble is translating your own LL-37 is important into administering LL-37 as a drug helps. Those are completely different claims, and the second one has very little behind it. What actual interventional trials exist? The most developed is topical for wound healing. Because chronic non-healing wounds tend to be low in LL-37, researchers ran early phase trials applying LL-37 topically to hard-to-heal venous leg ulcers. These were small, early-phase, dose-finding studies. LL-37 was well tolerated on the skin, and there was a signal of improved healing. But tellingly, it was dose-dependent in a way that should make you cautious. The benefit appeared at lower doses, and the highest doses did not perform best. That hints at exactly the double-edged character we've been discussing, a therapeutic window, above which the same molecule may stop helping. Beyond wound healing, there's been early exploratory work injecting LL-37 directly into tumors, particularly melanoma, as an experimental immunotherapy, very early stage. And there's been a small study delivering it orally via engineered bacteria in a respiratory infection context, which was tolerated. That is essentially the human interventional record, narrow, early, mostly topical or local, mostly about infection and wound healing. What there is not, any completed controlled human trial supporting systemic, injected LL-37 as an immune booster, a longevity intervention, or a general anti-aging therapy. No established systemic dose, no long-term human safety data for that use, no demonstrated anti-aging efficacy of any kind. And I have to be blunt here, more blunt than usual, because of the double-edged biology. There is a gray market selling LL-37 as an injectable, marketed for immune support, for chronic infections, for Lyme disease, for biofilm-associated illness, and yes, increasingly, for anti-aging. Someone offering you injectable LL-37 with those claims is operating far ahead of the evidence. But it's worse than that, and this is the part that separates LL-37 from the merely unproven compounds I've criticized in previous episodes. With most unproven peptides, the main risk is that they don't work and you've wasted your money. With LL-37, you are dealing with a molecule that is a validated driver of psoriasis, a validated autoantigen in lupus, a central player in rosacea, and a context-dependent modulator of tumor growth. In someone with a predisposition to any of those conditions, and many people carry such predispositions without knowing it, flooding the body with exogenous LL-37 is not a neutral gamble. It has a plausible mechanism for harm. That's not a hypothetical objection. It's the same biology I've spent half this episode describing. The regulatory picture. This is the point in every episode where I tell you what the regulatory reality actually is. And for LL-37, it's unusually concrete, which I appreciate because it means I'm not just offering an opinion. In the United States, the FDA has placed LL-37 on what's called the Category 2 Bulk Drug Substances list. In plain language, that's the list of substances the agency has reviewed for use in compounded medications and flagged as raising significant safety concerns, substances that should not be compounded into products for human use. So this isn't a peptide sitting in a gray zone of nobody's looked at it. A regulator has looked at it, specifically in the context of people wanting to compound it into injectables, and concluded there are safety concerns serious enough to keep it off the permitted list. That is about as clear a regulatory signal as this field ever gives you, which means the injectable LL-37 being sold is by and large sold under the research use only label, not as an approved or even a legally compoundable human medicine. When you see that label, understand what it means. It means no regulator has cleared this for you to put in your body, and the double-edged biology is precisely why. Where does LL-37 fit in aging at all? Let me address the question you might reasonably be asking. If this isn't a longevity peptide, why is it on a longevity list? Honestly, because the peptide longevity world has adopted it, and I'd rather cover it with rigor than leave you to the marketing. But there is a legitimate, if subtle, connection to aging, and it's worth stating precisely because it's not the connection the sellers imply. The sellers imply the mitochondrial peptide story. Levels fall with age, so top it up. That's not the real link. The real links are three, and they're all more complicated than boost it. First, immunosenescence. As we age, innate immunity becomes less effective and susceptibility to infection rises. Antimicrobial peptides are part of that front line, so a well-functioning cathelicidin system is part of aging well from an infectious disease standpoint. But that argues for keeping the system healthy, not for injecting the peptide. Second, vitamin D. Vitamin D insufficiency becomes more common with age, less sun exposure, less efficient skin synthesis, more time indoors. Since vitamin D drives the LL-37 gene, an older person who is genuinely vitamin D deficient may underproduce cathelicidin when challenged. Correcting a real deficiency is sensible. That's the actionable piece. And notice it's about nutrition, not about the peptide as a drug. Third, and most importantly, inflammaging. One of the central features of biological aging is chronic, low-grade, sterile inflammation. Inflammation without an infection to justify it. And LL-37, through exactly the self-DNA and self-RNA mechanisms I described, is a molecule that can convert your own dying cell debris into an inflammatory signal. In other words, in the context of aging, LL-37's more relevant role may not be as a protective factor you're missing, but as a potential contributor to the chronic inflammation you'd want to avoid. That completely inverts the top-it-up logic. It's the strongest reason to treat this peptide with respect rather than enthusiasm. What then is sensible? So, if the injectable is off the table, and for LL-37, I'm telling you plainly, it should be off the table for anyone without a specific medical reason and specialist supervision. What does a sensible person actually do with this information? The answer is boring, and it's the same answer that keeps coming up in this series, because the boring answers are the ones with evidence behind them. Support the system, don't flood it. Ensure your vitamin D is adequate. If you're deficient, correct it, because that's the genuine, mechanistically grounded lever on cathelicidin, and it has plenty of other benefits. But repleting a deficiency, not megadosing on top of sufficiency. Beyond that, the general levers of innate immune health apply. Sleep, which is when a great deal of immune regulation happens. Regular physical activity, not being chronically metabolically overloaded, and adequate protein and micronutrient status, including zinc, which supports antimicrobial peptide function. None of that is exciting, and all of it is defensible. And there's a caution specific to this molecule that I want to make explicit, because it doesn't apply to the others. If you have psoriasis, rosacea, lupus, or any autoimmune or chronic inflammatory condition, or a strong family history of one, LL-37 is a molecule you have specific reason to be wary of, not to seek out. For you in particular, the double-edged biology isn't an abstract concern, it's your disease mechanism. Where this leaves LL-37 in 2026. Let me close the way I always do with the honest summary. Conceptually, LL-37 is a beautiful piece of biology. It's your body's own antibiotic, a single, elegant, positively charged helix that kills a huge range of microbes on contact, neutralizes endotoxin, calls in immune cells, heals wounds, and links your innate immunity to your vitamin D status. As a piece of evolved host defense, it's genuinely impressive, and its endogenous importance to human health is one of the better established facts in this whole series. We know it matters, because people who lack it get sick. But as an intervention, as something to administer, to inject, to raise, it sits in a very different place from where its marketing puts it. The human interventional evidence is narrow. A few early topical wound healing studies, some exploratory local oncology work. There is no controlled evidence for systemic LL-37 as an immune or anti-aging therapy, no established dose, no long-term safety data for that use. And uniquely on this list, the molecule carries a well-characterized capacity for harm. It is a validated driver of psoriasis, a lupus autoantigen, central to rosacea, and a context-dependent modulator of tumor growth. A regulator has looked at it specifically and flagged it as a safety concern for human compounding. That is not a compound where the honest recommendation is levels fall with age, so top it up. The honest recommendation is respect the biology, support the system through vitamin D sufficiency and general health, and be especially cautious if you carry any inflammatory or autoimmune predisposition. So on the spectrum, this series keeps returning to, from credible research target to clinically actionable, LL-37 occupies a strange dual position. Its endogenous biology is more firmly established than almost anything we've covered. Its case as an injectable intervention is weaker and riskier than almost anything we've covered. Both things are true at once, and holding both in your head is the entire point. That's LL-37, the only human cathelicidin, named without romance for what it is. 2 leucines and 37 residues. Your own broad-spectrum antibiotic, stationed at every barrier where your body meets the world. A superb defender when it fires correctly, and a documented arsonist when it doesn't. The one peptide in this series where the honest advice runs opposite to the marketing. Not a molecule to chase, but a system to keep in good working order. In the next episode, we'll turn to a peptide from a completely different world. One that the internet has fallen even harder for than this one, sold everywhere for injury, recovery, and gut health, with an evidence base that is fascinating, mostly preclinical, and wildly out of proportion to the confidence of the people selling it. We'll do it the same way. What it is, what the proposed mechanisms are, what the evidence actually shows, and what the honest picture looks like. If this series is useful to you, share it with someone who's navigating this space. This field deserves more rigorous public discussion and less noise. I'm Dr. Ethan Hausman-Markey. This is The Science of Ageless Living.